Hi guys, my name is Dakota Tedder, and for my final project for Bio 111, my topic is stem cell research for type 1 diabetes. So a brief introduction of my topic. What is type 1 diabetes? Type 1 diabetes is triggered by an immune-mediated assault on insulin secreting from the pancreas cells. Type 1 diabetes mellitus is the most common autoimmune disease among individuals. It is the main symptom in the loss of pancreatic cells. That's the main symptom, is the loss of pancreatic cells, which results in insulin insufficiency and hyperglycemia. Exogenous insulin cannot be given or injected to replace the endogenous insulin produced by a healthy pancreas. MSCs are an interesting therapeutic alternative when compared to other types of stem cells because of their immunology properties and ability to differentiate in vitro given birth cells that generate insulin. And in the next few slides, I'm going to explain what MSCs are. So this is a very brief slide on the history of stem cell therapy and a little bit of early research. Stem cell treatment offers a lot of promise for treating T1DM patients. With the advent of research on this therapy for many disorders, there has already been progress in stem cell-based treatment for T1DM. A little bit of history continuing here. Harvard researchers used stem cells in treating type 1 diabetes in mice with success. Many unresolved issues must be addressed before this therapy can be used to treat diabetic patients. The team developed the HCELL homing molecule to direct stem cells to inflamed islets in the pancreas, or you can say H cell there. We'll try to switch the slide. Here is the MSC, what it means, and I'll butcher that word if I say it, so you can read it on the slide. Okay, and here is the MSC in treatment for T1DM. In 1976, Friedrichstein and colleagues obtained the first colonies of cells resembling fibroblasts from bone marrow. Because of their features, Chapman named those cells the MSC cells. That word I'm going to butcher. The BMMSCs are multipotent, non-hematopoietic stromal cells that can attach to cell culture surfaces and exhibit long-term differentiation and self-renewal. Nevertheless, the cell type is currently referred or called MMSC, diabetes mellitus type 1. That is what T1DM means. Results in cell death and reduced insulin production. I'm sorry guys, I'm going to butcher that. MSC. We'll wait for the next slide. The next slide is macrovessels derived from MSCs. So that's the M there. So macrovessels, also called MVs, are macroparticles that let cells communicate with one another. They are released by various MSCs and cell types. It was recently shown that MVs generated from MSCs CD44, CD73, and CD29. Recently, it was shown that MVs generated from MSCs had regenerative effects in a range of disease types. So the application of pluripotent stem cells in treatment of type 1 diabetes. Blastocyst walls are used to harbor pluripotent stem cells. IPSCs have an unlimited proliferative capacity as well as self-renewal, and they can develop in vitro and numerous adult stem cell types. The IPSCs are mostly produced from reprogrammed somatic cells, may develop proliferate in the same way that IPSCs do. Wait for the next slide to pop up. So the application. As a result, the researchers decided to see if the immature cells derived from HESCs might mature into fully functional cells of vivo. They employed an enhanced procedure to generate pancreatic endoderm cells with properties identical to the pancreatic tissue from a 9-week fetus, which they subsequently transplanted into test animals. Mostly they did mice and rats. The conclusion. MSCs are promising target for a variety of gene treatments used to treat a wide range of illness. They appear to be intriguing possibilities for delivering genes as well as repairing tissue and organ functions due to their apparent strong self-renewal capacity. The ability to generically design MSCs enables the expression of therapeutic genes over a lengthy period of time. And last but not least, the second part of my conclusion is further clinical studies using MSC sources. A greater number of patients getting transplants and a longer observation period are required to evaluate the effectiveness and safety of the novel transplant agency. Furthermore, future study must examine the importance of biomaterials, growth factors, and improvements in oxygen transportators. Okay, thank you for sticking with me while producing or trying to butcher more these words.